Psilocybin from a biochemical, a pharmacological, as well as a physiological perspective. Um, and the reason I chose psilocybin, I want to make more videos like this, but I chose psilocybin just because it's getting so much attention in the mainstream. It's got just a really interesting history, being a organic natural substance that's been around for quite a while and used for quite a while. But now it's really just shifted into the medical mainstream and getting a look in psychiatry and changing its legal status and all this stuff. So, without further ado, we're going to go over a quick history of psilocybin. So, psilocybin mushrooms refer broadly to a collection of fungi that are really loosely related. Um, they're not siblings, so to speak, from like a biological evolutionary genealogy standpoint. They're more like cousins within the fungi world, but they are classified mainly based off of the fact that they all contain psilocybin, the active or the psychoactive component of those mushrooms. And so, if we look at kind of the anthropology of this, there is some evidence, people think, of prehistoric consumption. People have cited cave rock art that looks, like they're worshiping a mushroom, or there's figures with mushrooms around, and they seem to think that that's evidence. I'm sure some people have also heard of the like stoned ape theory, which basically thinks that some psychoactive drug was responsible for um, primates and humans event, or the primate human segue, and that psychoactive drugs had a kind of catalytic role in that. Um, and then, in a more well documented sense, Mesoamerican use is very well documented. The Aztecs called, I'm going to murder that name, I'm not going to even try, but they basically called the mushrooms the god's flesh, and they were used in religious and healing rituals. And then, fast forwarding all the way to 1952, the modern, quote unquote, modern story of psilocybin picks up with mycologist Gordon Wasson, and he visits Oaxaca, Mexico, and participates in a mushroom ritual with Maria Sabina. And that was a pretty notorious feature in Life, or led to a notorious feature in Life magazine. And of note in this situation is, you can imagine that this interaction and his experience led to a lot of notoriety going to Maria Sabina and to the culture and to psilocybin in general. And Maria Sabina was quoted saying that she regretted facilitating this and allowing someone into the ritual. I think the idea there is with the kind of adulteration of the process, appropriation, and just the fact that something that was uh, important to them was kind of commercialized after that, not long after. In 1958, Albert Hofmann, a chemist who is pretty famous for discovering LSD, and his experience with LSD is where Bicycle Day originated. He does some chemical work and identifies that psilocybin and psilocin are the active compounds in magic mushrooms. In 1960, Timothy Leary and Richard Alpert, aka Ramdas, start the Harvard Psilocybin Project, and they perform the Good Friday experiment, which is where 20 or so divinity students were divided into psilocybin groups and non-psilocybin groups, or blinded, quote unquote blinded, although I don't know how you blind with something as potent as psilocybin. But nonetheless, they did that, and you can kind of imagine the chaos that followed. This is also just us on the doorstep of the 60s, the counterculture movement, the anti-war movement. And so this kind of unfolding was just really stirring the pot, and it got a lot of attention. And then on the other end of the decade, in 1970, we all know what happened after that decade is the war on drugs was enacted. Psilocybin becomes one of the many Schedule One controlled substances and kind of scapegoats, if you will, of Nixon's kind of whole public enemy number one ordeal. And Then we have a long hiatus. The stigma, the criminalization, all that stuff, obviously is going to push this underground. But even then, things were somewhat limited. Um, there is obviously clandestine stuff going on, and I'm sure we've all heard stories of the different subcultures in psychedelia or psychedelics. But things enter the limelight of the mainstream, or re-enter, I should say, in the early 2000s with Roland Griffiths, a really well-respected researcher at Johns Hopkins School of Medicine. And he's actually cleared to resume research on psilocybin as a potentially medicinal / psychiatric intervention. The medical field, I think, had just been shifting for a while. New research, new kind of ways of exploring consciousness, and things like SSRIs, antidepressants, and looking for those pharmacological ways of helping people's mental states. I think just led people kind of full circle. The culture changed a little bit, obviously, with legalization of weed and whatnot. I think Nixon's war on drugs and the system set in place from that were starting to get re-examined, and maybe they were even eroding a little bit. And so going into basic chemistry, here on the left, we have psilocybin in the flesh, or I should say in its nice little chemical structure representation. So of note, kind of scaffold starting or precursor, I should say, is really, really common or recurrent in biochemistry. If we were to look at the amino acid tryptophan or tryptamine, basically a very similar starting point here. It's no wonder that in our own biochemistry, that serotonin, this molecule right here, very well-known neurotransmitter, is derived from tryptophan. Some of you might have heard of that before. And so you can maybe see kind of where this direction we're going here is that psilocybin has this big phosphate group, which makes it polar, which makes it, not the easiest to get into the brain and start acting, because polarity is an enemy of the blood-brain barrier. But in the body, psilocybin undergoes a quick transformation of psilocin here. And you can see how this is a much bigger group, avoiding some of the chemistry. Basically, this transition makes it less polar. It's easier to transfer into the brain. And then that makes sense as far as its action in the brain or how it's able to get into the brain, because it resembles what we have in our body already, serotonin, to a pretty drastic extent. The difference mainly being that right here we see these two, this is signifying two methyl groups. So there's some bulkier groups up here, which is going to change. Chemistry is very nuanced. It's going to change binding. It's going to change how it acts, duration, van der Waals forces, things like that. But ultimately, the idea is that, there's a little bit of, there's an analogy here going on. So yeah, this is just a little bit more information on that. Psilocybin is a tryptamine alkaloid, some fun nomenclature, chemical nomenclature stuff. Psilocin is dephosphorylated. We talked about that. And what it's dephosphorylated by is alkaline phosphatases and nonspecific esterases in several areas of the body. So intestine, kidney, perhaps in the blood, to generate this compound psilocin. And the reason that's interesting is just, I always think, people kind of treat drugs like potions, but it's cool to know that, it is chemistry at the end of the day, and to a large extent, and it's going into your body. Stuff's happening on it. Your organs and different enzymes are acting on it, and it's getting metabolized. And so that's going to come into play later with things like half-life and whatnot. But just thinking about, the underlying stuff and processes that are undergoing your body, or the body during these things is very interesting. Okay, shifting into pharmokinetics, which is how your body acts on the substance, opposed to pharmacodynamics, which is the opposite. But psilocybin is converted to psilocin. We discussed that already. But the term for this conversion and the constant metabolism in that first first act, so to speak, is first pass metabolism. The small intestine's liver's a huge player in first pass metabolism. There's a connection there called the hepatic portal system. And the idea is that a lot of the things absorbed in the blood from your digestive tract is kind of going through a safety check first with the liver. A lot of metabolization happens here. Some stuff is even excreted after this metabolization relatively quickly. But some of these things, you can imagine there's kind of reactions going on. They survive for a little while. And then we get into this idea of, the timing, the half-life, and what's going on and how long. The onset of effects, loosely speaking, are usually 20 to 40 minutes. Psilocin in plasma, I think, peaks around 30 minutes. We start seeing psilocin in plasma around 30 minutes. Peak concentration of psilocin doesn't happen until about 2 hours after consumption. And then half-life, which is the time starting for the starting concentration of psilocin or psilocybin to decrease into half, makes sense, right? Half-life is equal to 1.5 to about 2.5 hours. So this gives you an idea of if we were to kind of see a little graph of how this stuff is peaking in the blood and then how it's kind of slowly, trickling out. We can get an idea for the curve, how long this stuff is really in our body. And then, this stuff, pharmacology, this is of, utmost importance for things like when we're dosing drugs in like a medical setting. But for anyone who's indulging in a recreational sense, I mean, not that this video is condoning that, but if you do, it's just helpful to think about the half-lives and the kind of getting under the hood of the body and what this stuff is doing there, just so you can, maybe make more informed choices. We already drew an analogy between psilocybin and serotonin, or psilocin and serotonin. But it's of note that they are both metabolized by demethylation, which is a chemical process, and oxidative deamination that is catalyzed by liver monoamine oxidase. And so MAO, or for short for monoamine oxidase, or aldehyde dehydrogenase, both enzymes. These enzymes can sometimes be taken, the inhibitors of these enzymes can sometimes be taken to slow drug metabolism and enhance effects. This is also seen in like drugs like DMT, where the taking of inhibitors for the enzymes that break them down, big surprise, slows down how you break them down, right? So if something's deconstructing the drugs you're taking and you inhibit that deconstruction, you're going to have the drugs in your system for a little bit longer. The conversion pathway, this is some of the known pathways. So psilocybin is psilocin, psilocin to 4-hydroxyindyl 3-acetaldehyde, fun stuff, big long names, more big long names. Of note is this right here in this left part of the figure, glucuronide, I always murder that. The process of glucuronidation, I always murder that word, but this big group added here, this is even bigger than the phosphate group that we had on earlier, the phosphoryl group. And so you can imagine how, I'll skip to the chase, but these oxygens and hydroxyl groups make this, and even this carboxyl group here, it makes all this really polar. And polar, there's kind of a loose, or I should say a pretty strict trend actually, of polarity helping with excretion and also preventing uptake into the cells, because non-polar stuff tends to kind of go through membranes and get deposited or distributed throughout the tissues, where polarity can sometimes keep things aqueous and keep them on their way, on the kind of plumbing highway, or on the plumbing out, on the way out. And so next we have site of action. So we're talking about chemistry, and then we're thinking, okay, well, if chemistry is going on, where is this chemistry going on in the body? And so there's metabolism going on, there's stuff changing, but where do we think these things are acting to cause all the crazy effects we have? And site of action is a really key, interesting concept, because whenever we have a drug in the body, it might act, we like to think, oh, it acts at the brain, or if you're spiritualizing these things, you might think, it acts kind of in some metaphysical way, and that's cool and all. But it's also good to know that, drugs do a lot in the body, not intended. And so this is where side effects come in play, because side of action can be very specific, but depending on the delivery, depending on the metabolism, depending on where the site of action is, it can sometimes be acting at multiple places. The most characterized site of action for psilocybin is the 5-HT2A receptor. This is a serotonin receptor. 5-HT is short for the chemical name for serotonin. But there's also other receptors where we think psilocybin acts at. One such receptor is the tyrosine receptor kinase B. This receptor has some role in neurotrophic or like brain neuron growing dynamics. So we'll get into that a little bit later. That's maybe a little bit of a misleading thing. It's not just like it, makes you super smart, big brain, but it does some interesting things in the brain, and we'll revisit that later. 5-HT2 receptors are a family of G-coupled protein receptors. And so this is important because what ultimately does binding of psilocin or serotonin at these receptors do? Well, in the case of psilocin, we'll get into that a little bit later, but it does bind at G-coupled protein receptors. These receptors broadly bind a ligand, e.g. psilocin. In this case, a ligand is usually a chemical that's just binding to these receptors. It enacts a cellular signaling cascade. So it binds here. This is at the cell membrane. On the inside of the cell, it's going to cause some conformational shifts of the protein. The protein's going to, because this is a channel protein here, it's going to have some effects, usually with like an ATPase or a secondary messenger amplification thing. Don't need to worry about that. Long story short, it causes a cascade that changes some cellular response. And so there's a lot of G-coupled protein receptors. That's a broad family, but 5-HT2 receptors are a subfamily within that. And these are the ones where most of the classical hallucinogens of interest are binding at. So LSD binds for a very, very long time, meaning it kind of hugs onto that. And I'm not even sure if it actually unbinds or if the receptor is internalized and metabolized. I'd have to review that. But mescaline also binds here. And so it's just an interesting kind of convergence of how these things are acting from a chemical standpoint. Multiple signaling and response pathways are involved. So this cartoon doesn't really do it justice. Signaling of this sort is super complex in any drug or anything. And so side effects, what's even happening that we maybe don't know fully and how well characterizes that. That's an ongoing area of research in a lot of ways in pharmacology, is looking at cell signaling and the different pathways that are involved with drugs. This is taking it a little bit more, the previous representation of the protein a little bit, and the receptor a little bit deeper. So this is still pretty cartoony by maybe chemist standards, but basically this is showing you the theme of the protein. So these little ribbons are a motif in proteins called alpha helixes. Situated here in yellow that's the psilocin right there. So you can imagine it would come in and it would bind and interact with these different chemical structures. And I like this just because it kind of gives you an idea for how chemical this stuff is, right? Like, again, not just a potion, not just like some liquid that goes into you and makes you feel all woozy. It's broken down and there's different ingredients in there, but ultimately psilocin is really formative in this process. And if you're wondering how they know that stuff, a lot of the studies are looking at if they block, psilocin from doing this whole choreography with these receptors, do you still have the trips and the crazy effects? And a lot of times you don't. So you can argue, it's a philosophical thing to talk about, if all there is to the crazy experience is chemistry. I'll save that for neuroscientists and philosophers and people, on these substances late in the night talking about stuff. But it's just cool to know that there is a lot of chemistry that's very essential. Ultimately, it's necessary for these things to have the effect they have. And so here's on this right side, we have psilocin in that binding pocket with all these, you can see all the crazy amino acid residues that in the protein chain here that are forming around this. And when psilocin comes in and the binding changes, this is the stuff that's refiguring and interacting with to shape cellular responses. I'm continuing on the side of action. So where are those proteins found in our body? Well, the 5-HT2A receptor is mainly, in this context, is mainly the area that we're mainly interested in is the brain, right? This receptor is found in all layers of the cortex of the brain, highest concentration being in the layer 5 pyramidal neurons. These neurons are very important in integration. They have their name because they have this kind of like upside down pyramid look where they have all these dendrites, which are the receiving end of neurons, branching out almost like a tree, right? And they have all these inputs coming in and this integration and this idea of sensory processing or signal processing is going on. And in the cortex, we also know that the 5-HT2A receptor is found on astrocytes. So that's another, what they call glial cell. It's another cell type that supports neurons. It has a lot of other roles it's doing, and that's an ongoing area of research as far as does it even help with signal conduction and other things. And I think it's just super fascinating that there's so much to be learned here, but there's also some really cool stuff being discovered inside our heads. 5-HT receptors, and this is really actually important, they have significant roles in a lot of different things in the body, so not just in the brain. The reason I think this is so important is because they have a role in nociceptive, so pain and motor function, and they're found peripherally, which is basically a way of saying not in the central nervous system, aka your brain and spine, but in places like the GI tract, involved in bowel movements. And this means ultimately that psilocin doesn't just act at the brain level, and that when you take drugs, just like when you take any drug, that even if it's natural and it's from nature and it's awesome, there's, and that's not even getting into the naturalistic fallacy, but that there's side effects and that these drugs have off-target effects. They act at different things. They might slow down your bowel movement. They might speed it up. They might sensitize you to pain. They might desensitize you to pain. They might slow motor reflexes or things of that nature. And not that any of these things are inherently bad, but they're important because, again, no drug is going to be a complete, perfect panacea. It's not going to, as long as it has a physical nature and acts with chemical constraints, it's going to have some pluses and some cons, or some pros and some cons. And maybe those pros vastly outweigh the cons, and that's great. And I think a lot of recreational substances that people enjoy, they find that's the case. But it's just good to know. And so now shifting away from the chemistry and the biochemistry and the pharmacology, more into kind of the physiological, psychological side, let's explore some early theories that people had about how the brain, I love this part because it's kind of talking about the philosophy of mind. And that's why I think psychedelics are kind of interesting, is they give us a little bit of an avenue to explore the psychology of mind and tell us a little bit about ourselves. But people obviously have been discussing the mind for a very long time, and independent of whether or not people were tripping out on or indulging on psychedelics. An early quote from the 18th century is by William Blake's The Marriage of Heaven and Hell. If the doors of perception were cleansed, everything would appear to man as it is, infinite. For man has closed himself up till he sees all things through narrow chinks of his cavern. So it kind of sounds like allegory of the cave a little bit, right? Narrow chinks of his cavern. Kind of, draws into question this whole thing of what's the limits of our perception, and do we really see everything, and how objective really are we in our self-proclaimed empiricism? And so, that could be explored more, but this kind of kicks off this literature around psychedelics. One key figure in the 20th century literature is Aldous Huxley, and he kind of recycles and transmutes this quote above and uses it for the title of his own psychedelic account when he was taking mescaline. The book's called The Doors of Perception. I definitely recommend you reading it if this stuff interests you. It's lucid. And just coming from a literary figure, I think it's interesting to see, someone that has that mental lucidity write about something that people generally think is disorienting and, kind of, it's stereotyped into being something that doesn't really help your fidelity and your reportage and your ability to kind of be in touch with yourself and reality. I think people sometimes get this feeling like, these things tear you away from reality. And whether or not that's the case is not the point. The point being that, when someone can write intelligently on something that I think is a very deep subject matter, it's compelling. And so Aldous Huxley does that, or explores that in The Doors of Perception. Huxley, a known syncretist, meaning he takes, he's synchronizing a lot of different things and taking a lot of different things and kind of blending them together. He, in his book, adopted this kind of Blakean idea of things being infinite, but us getting only a limited picture of that. Henry Bergson, a French philosopher of mind, is also known for thinking something similar, that we kind of have a valve that we, can only pay attention to so much. And this is relatively accurate to some modern conceptions of the brain, being that, we have limited attentional resources, but we have a lot of information that we're processing at any given moment. This idea of, before somebody even walks in the room, the smell, the nonverbal, or before they speak, the nonverbals, the smell, all the things that we might not be focusing our attention on, we're still receiving that. And our brain, or deeper parts of our brain, are deciding whether or not that information is worthy of attention, or if it's salient or not. And so Huxley, Blake, Henry Bergson, and others were a bit ahead of their time in thinking that psychedelics cleaned and loosened the nozzle of the sensorium, aka it allowed our senses and mental perception to be less picky, be looser, and kind of more accepting and inclusive. And given the brain's privileged role in physiology, since these accounts started building, the main effects in current research around psychedelics has been centered on the neuronal and cognitive changes we see when we take these substances. And so, thinking about how the physiology and psychology relate, right, being two sides of the same coin in a lot of ways, the psychological intensity of an experience, as reported through, subjective questionnaires or surveys, or also just, guided experiments with people that were administered these drugs, or maybe even animal models where they have behaviors that they associate with psychological intensity, and they can look at those animals and look at their blood levels in real time, or not real time, but relatively, really in a relatively tight timeline. And what they'll find is that the psychological intensity does correlate with the 5-H2A receptor binding that we talked about previously. So basically saying the more drug that's in your bloodstream and the more that it's binding, the more intense the psychological experience. That makes sense. But that goes along with this idea that there's some very physical, chemical stuff that's kind of underlying, or at least necessary for the psychological and even maybe the spiritual aspects to commence. Psilocybin disrupted connectivity across cortical networks and subcortical structures. And so what was found in a lot of studies is that when you take psilocybin, there is, your brain has kind of more established highways of connection between different areas, doing different jobs of processing and, diffuse, or what some people use as sparse representations of different things. And then you have subcortical structures, meaning not in the kind of outer area of your brain, but in the below that, the subliminal part. There's all these connections, right? And so psilocybin desynchronizes some of that. And part of the idea too is that there is more connectivity amongst parts that are not normally connected, meaning, or where the activity is not quite as intense usually. And then there is less kind of functional connectivity between networks that are usually our defaults. This desynchronization was observed across association cortex, so that's a kind of broad cortex of the brain, but was strongest in the default mode network. So this is interesting because this has been getting a lot of attention lately in the cognitive aspect of these studies, which is this network is been associated with our sense of self in certain tasks or in certain kind of more abstractions where people are kind of aware of themselves. This network is very active. And so the kind of rhythmicity of this, the kind of how this, the name kind of implies a little bit about that, but this kind of default mode network, how it kind of in our default mode, how this is operating, this is compromised to some extent when we take psychedelics. And so that kind of could maybe be a little bit of the neuroscience or the neuronal basis for kind of that dissolution of self that people experience when they take shrooms or other psychedelics. This also maybe hints at the basis for how, when you are under the influence of these things, doing some perceptual tasks, a simple perceptual task, can actually ground you. So, trying to pick up something and kind of focus on a simple object, real object, and connecting with quote unquote physical reality during psychedelic therapy has been shown to help some people if they're having a little bit of a, floating a little too high, flying a little too close to the sun experience. The grounding can be helpful. And so going back into maybe a little bit more of like a procedural or perceptual task, I'm speculating completely here, but I don't know if that goes into a more rudimentary part of the brain or just kind of gets things resynchronized a little bit. But that's an interesting finding there. These results suggest that the integration of functional connectivity in sensory regions and the disintegration in associative regions may underlie the psychedelic state and pinpoint the critical role of serotonin 2A and 1A receptor systems. So this also could be, this connectivity that we're talking about could also be the idea of synesthesia that's been reported before in people that don't normally have it, in people on psychedelics. So the idea of two senses kind of bleeding into each other and blending into each other. And it makes sense that if our connectivity is getting a little sloppy, and some people enjoy that, then that you're going to get a little bit of blending of sensory integration as you're under the influence. And so now shifting into the physiological, psychological effects, mainly tolerance and addiction. Of note is that, again, this is not medical advice. This is not prescribing or recommending anyone does take these substances or doesn't. But it is of note that obviously these substances are still illegal. From a tolerance standpoint, taking psilocybin doesn't seem to studies have shown that doesn't seem to reduce sensitivity to future doses of psilocybin. So whereas some drugs, alcohol, maybe not to the most extent, but maybe more things like cocaine, you see downregulation, you see your brain, change some of its mechanisms. So you eventually build more tolerance, and, that changes kind of the amount of drug or dosing that you need to then have a similar effect next time around. And that's obviously a little bit concerning when you need to keep, increasing your dose of a drug. That's not a good trend in any drug that we're interested in, whether it's legal or not legal. But tolerance is actually seen to a surprising amount with LSD. That might be just because LSD binds so long and is pretty damn potent. But crosstalk intolerance is actually still seen between LSD and psilocybin, suggesting that if you were to take LSD and then take a subsequent dose of psilocybin, you would actually have a lower sensitivity to psilocybin, meaning that, or that's kind of suggesting that there is similarity between the two and how they act and where they act, because clearly LSD can desensitize you to psilocybin's effects a little bit. And so if they were operating on two different independent tracks, that would not make as much sense. From an addiction standpoint, in animal studies, this is often looked at like, does the mouse push the lever again when the water is, tainted with cocaine or methamphetamines? I know, right? What you would give to be those rats. But in those studies and in just, studies that they've done with people in clinical settings, self-administration and reinforcing effects are not as commonly seen with psilocybin. Traditional reward behavioral pathway systems, such as the limbic system, are thought to be engaged really strongly, and that can be measured in different ways with, with animal models. Obviously, I think even with functional MRI and some other stuff, they can look at what systems and pathways are really active during different, during and post different ingestion of different substances. But these kind of traditional pathways are not really implicated as much with psilocybin, which for the idea of the upside of this as a psychiatric intervention is actually a good thing, right? We want a drug that can potentially help people in certain specific cases where it's medically fit, and we hope that the downside of that, as far as addiction or tolerance or side effects, is minimal. We covered this a little, or we touched on this a little earlier with the site of action, but we mentioned briefly that there's another receptor, the tyrosine receptor kinase B. And a lot of antidepressant drugs, including psychedelics, have some action here in addition to some of their other actions. The reason this is really interesting is that this idea of how can we combat addiction or how can we combat depression. A big idea is that you have these negative patterns of thought that are just hard to get out of, right? It's easy to tell someone, oh, just get over it, right? But if you're depressed and if the emotional valence in your head is dark and it's cloudy and you get stuck in a rut, it's hard to overcome the habits and the deeply ingrained you can even make an argument in circuitry that kind of evolves over time with mental changes. And so neuroplasticity is really interesting because this is kind of the hope is that, if we can kind of catalyze neuroplasticity in a targeted, beneficial, adaptive way, then you can help people kind of get out of those patterns by creating and displacing the negative stuff with newer, hopefully more positive mental circuitry. And so going back to messengers and receptors and how this works is that TRKB increases a thing called brain-derived neurotropic factor. This is something that facilitates neuronal growth and plasticity. It's common in the body. Things like exercise, a lot of things do this. So don't think that you have to just do drugs to get, healthy changes in your brain chemistry. But that in, certain drugs, antidepressant drugs, the idea is that this receptor has a couple different pathways it works through: the RAS mitogen activated protein kinase, or otherwise known as the MAPK pathway, the PI3K pathway, this other pathway. And all these effects have different things in the cell as far as transcription. Is basically saying, if we look down here, about after all this little signaling goes on, that something in the the core of your DNA is changing the creation of proteins, which are going to be sent out to the rest of the cell for different functional purposes. And so something kind of more permanent or lasting. Again, this stuff happens on a lot of different time scales. Some things are degraded very quickly. Some stuff gets integrated into the cell structure and has a little bit longer lifespan. But the idea is that something is happening that's more than just, it's lasting longer than the effect of the drug. That's causing some lasting change in your cellular biology. And so, interesting enough, there's a spinogenic effect of LSD. So that's this idea of that these little dendritic spines or things that can help, when axons are trying to connect with different stuff, that there's a little bit of a branching out effect going on here. Again, this happens more generally with BDNF in general, and most learning or most, formative periods of your life, there's neuroplasticity going on, right? So it doesn't just take a crazy drug to do that. But the idea is that if we can do targeted neuroplasticity, then there's, a way to go above and beyond what we're normally capable of achieving with just kind of, and people have, let's be real, people have barriers to, let's not be ableist. Some people have barriers to their ability to participate in different activities and physical activity and yada, yada. And so this idea is how can we get down to the bare bones of the chemical level without, the kind of lifestyle and all the big things that are the big limitations for a lot of people when it comes to improving quality of life. Actually, of note, going back to that real quick, is that the reason this pathway is also important outside the 5-HT2A part is that 5-HT2A receptors are known for the psychedelic experience. But a big area of research is that if we can target just the neuroplasticity element, which who knows if that's separable from the other pathway, this pathway, if we can focus on that, maybe, people that are not big fans of the hallucinations and whatnot are thinking that maybe we can kind of bias drugs towards neuroplasticity without all the extra hallucination and all that stuff. This is part of a big body of research called biased agonists, and that's a big topic, but it's a really cool topic looking at how can we continue to dial in pharmaceutical interventions, so they have less off target and they can, hopefully bias one pathway versus, setting off a million things that maybe we don't want all of those effects. But that you can imagine that's a very convoluted area to get into. Going a little further into this idea of plasticity, neuroplasticity is this base idea of, things changing on the neuronal level, but that's happening with good learning, good drugs, bad drugs. Not that good drugs and bad drugs exist, but quote unquote, different lifestyle things. Any learning or changes, substantial, lasting learning, usually plasticity is happening. But that's not what we're necessarily looking at. What we want to look at is our ability to change improved? And so if plasticity is improved just indiscriminately, that's not necessarily good. But metaplasticity is the idea of how good are we at getting more changeable. So how plastic is our plasticity? It's a little doubled up there. Don't want to try to confuse things too much. But if plasticity isn't inherently good and is happening in response to a lot of different stimuli, what we really want is the ability to facilitate plasticity when we want and adaptation when desired. And this goes after this idea of critical periods, which is this idea that, when our brain's developing, we have periods of being more malleable and more responsive to stimuli that can shape it. And so if we can, as we age naturally, we think that these critical periods close and that it's harder to learn, it's harder to change, right? The old dogs, new tricks type of ordeal. And certain things can help neuroplasticity, can maybe even help metaplasticity, such as, again, exercise, new experiences, yada, yada. But, if we can approach this from a pharmacological perspective, thinking back to this slide and then metaplasticity, so thinking how we can more globally allow for a little bit of loosening of the rigidness of our brain and our pathways and our way of functioning. This is one of the main goals of this whole thing, right? Is that if you're depressed or if you're in a bad place, change in variability is good, right? If you're in a bad place and you roll the dice again a little bit, even if you are rolling the dice a little bit, again, you could get a bad roll. And so that's something to think about, is that there is some kind of probability. There's a lot of things going on here, but it's also cool to know that maybe, just maybe, if you're in a really bad place, that as people hone in on these substances more, maybe there can be a way to facilitate adaptive rewiring and kind of combat that negative thinking. All right. And so that's pretty much it for this little exploration. I hope to make more videos of this nature. If you enjoyed this video, give it a like or a subscribe. This is on the Alkahest channel. We're going to be exploring more about pharmacology, drugs, consciousness, philosophy, literature around this stuff. And I'll see you next time.